I want to start out my talk by asking you a few questions which may be poignant to you. Are you tired of EHR research? Are you overwhelmed by the complexity of biobank phenotype data? Do you say to yourself, there has to be a better way?
Do you have your mic on?
No, I'm not mic'd. Thanks for stepping on my line. But there is a better way. It's Phecodes. Okay, so today I'm going to talk about Phecodes. This is something I use every single day. I've worked on Phecodes for 15 years, and I think if any of you are doing biobank research, EHR research, I think that there are ways that you can use Phecodes to make your work better. First of all, what are Phecodes? Phecodes are clinically meaningful groups of ICD codes that we've grouped together in order to capture certain clinical entities. As you guys probably know, if you're experienced with ICD codes, there are tens of thousands of ICD-9 and 10 codes. Many of those codes contain details that are not relevant to any of the studies that we want to do, like uveitis of the left eye, uveitis of the right eye, uveitis of both eyes, or uveitis I didn't specify. All of those say uveitis, so those are all grouped together in a single Phecode for uveitis, for instance. Phecodes also merge together ICD-9 and ICD-10 codes, and the version 1.2, which is the version that we froze all the way back in 2013 and have been using since, captures a little over 1,800 phenotypes out of these ICD codes. The Phecodes themselves look a lot like ICD-9s if you're familiar. They have this parent code, this 3-digit parent code, followed by decimals, which adds increasing specificity. So 0.11 would be a grandchild of 250, and 0.1 is the child of 250. We also, for Phecodes, define exclude ranges. That's for when you take a Phecode and you say, these are cases, and instead of everybody without Phecodes being controls, you say, we're going to exclude from controls people who have similar diseases. So in this example here, if you created a Phecode of type 1 diabetes with ketoacidosis for controls, you're excluding everybody with any diabetes code. That takes care of that. Yeah, so again, the whole principle of Phecodes is to take these ICD codes, which are nice structured data, and condense them in a way that's more useful for doing research. And as an example of the kind of meaningless detail that can be included in ICD codes, there's almost 500 codes for tuberculosis in ICD-9. It's one of the first codes that was created back when ICD structure was developed. And so there's basically nearly 500 ways of saying tuberculosis. If you were to use each one of those, serially in any sort of analysis, you'd be doing a lot of tests, or you'd be having a lot of dimensionality to your data that's really not very helpful. So again, Phecodes condenses all those into one. Phecodes are organized into chapters. These chapters are the same as the ICD-9 chapters, and which are very similar to ICD-10 as well. So you have codes for infectious disease, endocrine metabolic, your cancers, etc., all the way down to symptoms and injuries and poisoning. Phecodes are compatible with basically any I can't think of a biobank that Phecodes aren't compatible with. Why? Because ICDs are an international standard. They're set by the WHO. And all of these biobanks are linked to medical records, typically not clinical notes. You oftentimes can't get into the text of notes in things like UK biobanks, etc., but they are linked to ICD codes, all these patients. So Phecodes can be used. A quick note on this, though, in the US, just to get down to the nitty-gritty, we use something called ICD-CM. It's a clinical extension of ICD coding system that's a little bit different than what people in the UK, Japan, etc. use. And we have mappings for those that are available on our website. A very important, probably the most important aspect of Phecodes is that they actually appear to work. A lot of people expressed, especially early on, but still, a lot of skepticism that you can really get anywhere with just billing code data. After all, it's got this kind of filth kind of associated with it, this filthy lucre associated with it. It's just meant to extract money from people, and that is all true. There's a lot of incentives to give certain people certain ICD codes that is purely financial and doesn't have anything to do with biology, so there's reason to be skeptical. This study is probably the thing that was the study that I've most referred to of any of the studies I participated in mentally, because what we did was try to replicate all the associations that were in the PheWAS catalog at the time using Phecodes. And we successfully replicated a little over 2/3 of the associations that we were powered for, and these replications occurred across all different body systems. So we replicated cancers and neurological problems, cardiovascular problems, etc. That means that Phecodes aren't necessarily perfect. They could have some certain degree of noise, but that they're capturing very meaningful information across the medical phenome about these patients. So I'm going to quickly go through a few of the really super fun things that you can do with Phecodes. The first are phenome-wide association studies. This is what Phecodes were developed for. It was actually my boss at the time, Josh Denny, who came up with the idea of Phecodes. How did he come up with the idea of Phecodes? We were working on these projects to do EHR-based phenotyping in a thing called eMERGE, if you guys are familiar. It's a network of research hospitals that pool data together. And these algorithms were very, very complicated and used EHR data, and they were all rule-based. So for instance, you say somebody with hypothyroidism, we're going to require that they have a billing code for hyperthyroidism. We're going to require they're on levothyroxine because that's the treatment for hypothyroidism. We're going to exclude people who've had a thyroidectomy, and all these kind of complicated we're going to include lab data, medication data, problem list data, etc. They'd come up with these rules, right? And they did replication studies in these demonstration projects. But one thing that we noted pretty early on was that the common thread through all of these algorithms is that all of them used ICD codes. Some of them used EKGs, some of them used medications, but all of them used ICDs as a common data element as part of these algorithms. So then the idea came up, hey, maybe we could actually create phenotypes just using these ICD codes. And what that would allow us to do is do a high throughput capture of the medical phenome. And then we could do this thing called a phenome-wide association study. This is a total perfect corollary really with the genome-wide association study. But instead of looking at one trait across the whole human genome, all chromosomes, you take a single SNP and you say, what are the phenotypes that are associated with that single SNP? And this is a variant that we discovered in the eMERGE group doing a study on hypothyroidism with one of those fancy algorithms, and then we PheWAS'd it. So PheWAS was really a method to try to explore pleiotropy. It's still very, very commonly used. I think some people have presented PheWAS projects in this very session or week. So PheWAS, initially, we really developed the method to focus on common and complex disease. For that reason, we really focused on defining phenotypes that were for things like Crohn's disease or AMD, stuff that was in the GWAS catalog at the time. But the use cases of Phecodes and PheWAS has really been growing. Here's just a particularly lovely PheWAS plot. This came out of a study in Mount Sinai where they actually PheWAS'd a rare variant that's functional to describe sort of the phenotypic patterns of a functional variant in a collagen gene that was very common or more common than other populations in Puerto Rico. So it's not just for common variants. Oh, here's an important part about Phecodes. Okay, minimum code count. When we talk about minimum code count for PheWAS, you have a decision if you say, somebody has a Phecode, they're a case, somebody doesn't, they're a control, maybe use your exclude ranges. But what we noticed early on, and we never really officially published this except at a poster in AMI until I wrote this annual review thing. I hate to actually pump my own papers, but if you guys were curious about Phecodes, a couple years ago I literally wrote down everything I know about them for this annual review, so check out that paper. It's the best I could, I don't know if it's good, but it's the best I could do. What we looked at with this AMIA poster, which I reproduced this figure here because it's really important, is said, what's the positive predictive value of Phecodes, of having an ICD code for different diseases if you just require one code on one unique billing date, two unique billing dates, 3, 4, etc., right? And also, what's the negative predictive value? So with that, you can generate an F1 score. And what we found, and this finding has been reproduced in other contexts as well, is that requiring 2 or more unique dates for a case kind of hits this nice balance between positive and negative predictive value. Does that make sense, guys? The more codes that somebody has, the more likely it is that they actually have the condition. If somebody has if somebody's been billed for rheumatoid arthritis in 8 separate dates, they probably have rheumatoid arthritis, or at least their doctor thinks they have rheumatoid arthritis. If they only have one, that one billing code may have been used as a rule-out code. It may have been used to justify getting tests or something like that to see if they had it, etc. So this is kind of called the rule of 2. This is the default setting in this PheWAS R package, which I'm going to talk about here. So how do you, I'm sure you guys are just dying to use PheWAS right now if you haven't already. How are you going to do that? One way to do that is to use our R package. I'm currently in my lab. I have somebody who's working like full-time and getting this actually into CRAN, but we published this in 2013. It's a lot of people use it based on the number of times the paper has been cited. This is a very common package that people use to run PheWAS. What do you need to run PheWAS? There's really 3 ingredients. You need billing codes. You need some basic demographic information like birth date, sex. And then if you're going to use PheWAS to do genetics, which is a common, although not the exclusively the only application, you need some genetic data as well. And you put all those ingredients in here, and this package will mix them up for you and make you a PheWAS. Another way to access PheWAS, if you don't want to go to the trouble of running the package, or you don't have biobank data that you're working for, my friends at Michigan have done this beautiful job of making this website, which is called PheWeb. They have what we call a PheWAS by GWAS of the UK biobank data. That's free. Anybody can get it. And it's a beautiful browser that they developed. This programmer, Peter, he did such an awesome job here. And so here you can search for any variant, any gene, or any phenotype, and you can get your PheWAS data here, and it's all downloadable. Oh, we just released this. So as I was saying, we froze a version of Phecodes, the one that everybody uses, the one that's at that Michigan Biobank, at version 1.2. And that was what we published in 2013. When we developed those Phecodes, we were really trying to capture common complex disease, as I said. This is the stuff that people were GWASing at the time in the GWAS catalog. But that left a lot of detail on the table, okay? Like stuff that went down during pregnancy, let's say, preeclampsia. Maybe that wasn't in the GWAS catalog. A lot of congenital anomalies, which can be captured with ICD codes. Those codes were grouped into pretty general things, like congenital anomalies of the head, face, and neck, which would group together a bunch of ICD codes. So you weren't able to capture that detail. We started to increasingly rely on using Phecodes to do stuff in the Mendelian space, where very specific traits, congenital anomalies, of course, were very important, et cetera. So we devised a new version of Phecodes called Phecodex. This is before the Twitter thing, so now I'm kind of like feel a little funny about it, because I thought it was kind of cool at first, and now everybody's ripping on the idea. So, but it stands for Phecode extension. This may not supplant version 1.2, but this is compatible with the PheWAS R package. And you can go on GitHub and take a look at it. To do this, man, I'm telling this stuff out of order. Phecodes, we could have done something like, let's cluster these ICD codes together and let the machine kind of group things together. That is not what we did for Phecodes. We actually sat down with clinicians and said, what are clinically meaningful concepts? What concepts go together? How would you like this to be arranged? To do Phecodex, I personally met with 22 clinicians of different subspecialties. I've been at Vanderbilt long enough that I've worked with a lot of people. So I call up a friend in infectious disease, a neonatologist, et cetera, et cetera. And we actually, in principle, in a knowledge-based way, organize these codes. Okay, I've lingered on that long enough. One of the things that the new Phecodex has allowed us to do is to use this mom-baby database that we've put together to start looking at things beyond just genetic loci. So we might want to actually regress against a different type of variable. This is a study we recently published looking at bronchopulmonary dysplasia. That's a bad lung problem that babies who are born too early often get. A lot of the things of maternal risk factors, like during pregnancy, are not well understood how they reflect on babies. I mean, there's some stuff that we know, but there's like a lot of mystery there. So what we did was we said, what diagnoses in the mom during pregnancy impact the likelihood of having BPD in the baby? And the only reason we were able to do this because it was using Phecodex, because polyhydramnios and oligohydramnios, that's too much or too little amniotic fluid, those kind of features are not represented in version 1.2, so it adds a lot of detail. The real impetus for creating Phecodex, ooh, that's kind of blurry, was really to do this study, oh, and I have the wrong citation here, this is Chenji Zheng's study on hereditary cancer syndromes, which I presented on Monday, if you guys remember, which I don't blame you if you don't, it's been a lot. But again, this was a study looking at Mendelian variants or hereditary cancer variants, these rare pathogenic variants, and we simply didn't have the level of detail we needed in the version 1.2. So if you're interested in using Phecodex, Again, it's integrated into the R package, which is on GitHub. You can also go to our website. We have a lot of PheWAS related stuff, phewascatalog.org, and there'll be an explanation there. Okay, next fun thing you can do with Phecodes, very briefly, you can make phenotype risk scores. So as I presented on Monday, you can use these phenotypes because you really get the capture of a medical phenome and start looking for Mendelian disease patterns using these things. So we have created a map, and that is also published and downloadable, of Phecodes to HPO terms. Anybody who does Mendelian disease, you're probably very familiar with HPO terms. So we have a nice linkage there. And also, we provide that map in a new phenotype risk score package that Layla Aref has developed and actually got onto CRAN because she's like an excellent programmer. I'm not good at documenting things, so it's difficult for me to get things in CRAN. Okay, another really great thing that you can use for Phecodes, in addition to PheWAS and PheRS, and this one doesn't start with a PH, is to do cross-biobanking phenotyping. One thing when I was putting together a renewal for the PheWAS grant that I realized with a great deal of pride, I have to say, is that, oh gosh, what is this? The global biobank that people have been putting together, all these biobanks together, the number one way that they define phenotypes so that it works across all of these global biobanks is Phecodes. And so they're written right here, see Phecodes, right in their primary graphic. So Phecodes, because they're based on ICDs, are a way of actually being able to develop individual phenotypes that can work across biobanks, even if you're not doing a PheWAS, which why wouldn't you? The other thing here, this is a graphic that I produced just out of curiosity, looking at prevalences across different biobanks. I won't get into there's a lot of ways to interpret this, but the point is that as long as you have ICD codes from these different resources, this is BioView, All of Us, Michigan Biobank, and UK Biobank, you can compare the prevalences across populations, which can be a very helpful thing to do, and just basically profiling data. Oh, very quickly. So a lot of people use that PheWAS R package to make Phecodes, and I think I should put a tutorial or something up on our website because that's not actually the way that I use the Phecodes, and I just wanted to walk through this really quickly for those of you who like to use SQL, which I'm a big MySQL fan for data storage and basic data manipulation. Basically, I have a map table in SQL which maps ICDs, either ICD-9 or 10, which is shown here, to Phecodes, right? And then I have a table here that has ICD codes as a person ID. This is like one that got billed for it, what their ICD code, and that's again ICD-9 or 10. And then I run this query here, which creates a new table called Phecode counts. Ooh, I forgot to take a screenshot of it. But basically, it's a table that every person says if they have a Phecode and how many times that Phecode occurred. So basically, anytime I get a new data set, this is the very first thing that I do, is I generate Phecodes on all these patients. I generate prevalence data on it. And you know, a lot of times you'll notice something that's wrong. You know, you'll notice just by looking at the data in this way that there might be problems with the data or things that you want to know before proceeding in the analysis. I'm doing great on time. This is my last part. So one last thing that you can do with Phecodes that I think is super fun is to think about using them to do some basic quality control on data. So one thing that I've noted with a little bit of trepidation, because I realize that some of the work, at least that I participated in, has kind of moved us in this direction, is that when people really first started doing genome-wide association studies, like if you read that AMD paper, the age-related macular degeneration paper that was done in like 2005, in the early days, they were very careful about phenotyping. In that AMD paper, they looked at like the retinas or the eye pictures of everybody, had an ophthalmologist examine them, said, you're a case, you're a control, right? It was super, super clean. We've now moved to an era where we're really, not only are we not examining patients and enrolling them in certain studies to do GWAS, we're taking this very complex and noisy EHR data, and that's what we're using as phenotypes. Meanwhile, we've not really developed any strategies to do quality control on our phenome, right? So, and that's in stark contrast to when you think about all the wonderful quality control tools that you have for genome data. Like nobody in this room would want to say, I'm going to just use pre-QC genotyping data and just go ahead forth with your analysis. You know, instead there's all these allele frequency things where you try to match it to a reference population, Hardy-Weinberg, missingness, all of these statistics that help you know and that your genotype data is good and help you filter and clean your genotype data. And there really is no such corollary with the phenome. Part of the reason is that the phenome is like a lot more complicated. It's a lot more multidimensional than specifying variants in the genome. But one thing that we did, and this paper is about to come out at AJHG, but I think in September, but it's not quite out yet. We have a bioRxiv older copy, is to think about ways that we might be able to do some degree of phenotype quality control by looking at replication. And this is really harkens back to that 2013 paper where I said Phecodes work. Why did I think Phecodes worked? Why did I feel like that study demonstrated that? Because there's no reason, if Phecodes did not capture, let's say, rheumatoid arthritis and Crohn's disease, et cetera, there'd be no reason we'd be able to replicate those known associations from previous GWAS studies, and yet we do. So this idea is that in any biobank cohort of a decent size, which they all are at this point, there's a certain number of phenotype-genotype associations you ought to be able to replicate if all has gone well, if your data hasn't become corrupted, et cetera. And that if you're unable to replicate a lot of these associations, that may indicate some sort of data quality problem. Now, low replication in this context isn't going to point you exactly to what the problem is, but it's going to give you at least a yellow or a red light to say slow down, there may be something going on with your data because you're not seeing the patterns in this data that you expect to see. So maybe you don't want to go on and proceed to find new patterns and new correlations before you can replicate the old. To develop this idea of quality control through replication, we really thought about what were the kind of basic, what are we trying to do when we're trying to replicate things? Oh, I should mention this too because I think it's helpful. National Academy of Sciences did a really nice report on replication and reproducibility. It's a whole book. And maybe this is real 101 to you guys and I'm telling you something you already know, but they have a distinction between replication and reproduction. So reproducibility, reproducing your work is like you write the code, you run it on your data. You should be able to run the same code on the same data and get the same results. That's reproducibility. Replicability, on the other hand, is getting your results and then taking it to another data set and running the same experiment on a new data set and reproducing the results. That's replicability, okay? So I think that's actually a helpful distinction, although not everybody in every field abides by that distinction. But in replicability, the more I read about this, the more I realized there's kind of some deep philosophical waters in the idea of replicability that not everybody dives all the way into. Because in replicability, let's see if so what do we need to do with replication? We need another data set that's sufficiently powered, that's really easy, that use the same or similar phenotype. Okay, well that's already a little bit tricky. What do we mean by the same phenotype? Phenotypes are so multidimensional. For instance, I don't know how long ago, but maybe a decade ago, the definition of hypertension was changed, such that many, many more people qualified as having a diagnosis of hypertension. So you could have a 20-year-old cohort of hypertension, a 5-year-old cohort of hypertension, and then underneath the hood, there are actually pretty different phenotype definitions, but the same word is kind of put over it. We all call it all hypertension. So that's quite complicated, same or similar phenotype. Also, very complex, a similar population. What do we mean by that? Similar genetic ancestry, similar age, similar comorbidity patterns. What about the fact that, at our biobank in Nashville, we have a lot of metabolic things going on in the South, right? Replicating our is the UK biobank a similar cohort to our cohort in Vanderbilt? Got much lower BMI, they're older, they're healthier in almost every respect on average. And genetic model. This is a really nice write-up, by the way, in 2007 where they were thinking about replication. That I encourage you to read this paper. It's very clear. Although it's very clear, but they leave a lot of details to the imagination, I guess you could say. So what we wanted to do to create this, getting back to how you can use Phecodes to do this QC thing, we wanted to take the GWAS catalog and filter studies so that they met those criteria I just talked about for replication studies. So we filtered out everything that was, let's say, a modified phenotype. There's a lot of things in the GWAS catalog that's like BRCA1 positive breast cancer cases. Okay, that's not the same phenotype as breast cancer, so we checked those out. Or there'll be like type 2 diabetes in obese population. That is a kind of specialized population, so we dropped those studies out. Either way, we did a lot of filtering, but in the interest of time, I'm not going to go through it exhaustively. Really what we ended up with is what we call the Phenotype-Genotype Reference Map. This is, again, something that we publish. I'll put up the URL in a minute. And what the Phenotype-Genotype Reference Map is, these are all genotype, Phecode encoded phenotype associations with risk allele directions and effect sizes that are all in this resource, and ancestry annotations. And these are all associations that you can attempt to replicate in a new cohort, so long as you, and like I said, it works on all of these biobanks. We defined some replication measures. I want to leave time for questions, so yeah, I'm going to gloss over this a little bit because you guys might not be super interested in it. Basically, we tested this on a bunch of biobanks, found it worked really, really robustly. How can you use this Phenotype-Genotype Reference Map? Well, if you have a new data set, let's say you're going into biobank, like UK Biobank, and you're creating a special cohort, right? And you want to say, okay, did I do something wrong? I mean, if you guys are actually hands-on-the-keyboard data scientists, you know how often things go wrong, just in moving data around and things like that, and you want to feel like nice and secure about your data set. What you can do is use the PGRM package to just run those associations that are in the reference map. And then say, of those associations that I'm powered for at 80%, what percentage of them do I replicate, right? Well, we show, one of the things that we show in this study is that if you add noise to your data, so this is the Vanderbilt data set, you replicate just about almost 80% of those that you're 80% powered. But as you add noise, just random noise to your data set, it goes all the way down to 0, which shows that there's, even for this very crude replication measure, that it's sensitive to adding noise in the data. I should also point out, this part of my presentation is part of a longer presentation, but my friend called me the Smokey Bear of EHR research, which, like, I'm always cautioning people about, like, be careful, and I took that as a compliment. I'm not sure if it was, so I themed it out like that. I guess, like I said, I want to do questions here, but you know, if you look at our paper on bioRxiv, the one that's just going to come out in AJHG, you can see that there's a lot of scenarios where you can use replication as a way of testing different things. Maybe the cleanest example is here. For instance, remember I talked about that minimum code count? Here's the replication rate across all the biobanks when you change the minimum code count. So you say, okay, I'm going to count cases as having at least one ICD code on one unique date. For that, our replication rate is between 65 and 70%. Not that great, right? If you require 2, it jumps all the way up to a little over 75%, right? So you can see that increasing the minimum code count in our data set really increased it's decreased the noise and it increased the accuracy of the phenotype, which enables us to do more replication. And actually, really the higher the minimum code count sort of continues to get better, but of course you're losing power because by the time you require 8 ICD codes for your cases, you're throwing out everybody with 1 through 7 ICD codes, and you're losing a lot of cases there. But either way, we try the best we can to step through these examples in our paper, because I'm really, replication is something that for over 10 years, and I've been working as a data scientist, and it's something that I've done internally all the time. And I'll tell you guys, it's helped me catch some really major errors that made me really glad that I didn't proceed with and do a whole bunch of work on data set that had really major problems. So every time I get a new data set, I actually, far before we developed this PGRM thing, I had kind of a prototype version of it, and I would always run a replication study on new data sets. And I found, the one problem I will say, warn you if you start using the PGRM, is that you may have to deliver bad news to people, and nobody ever likes that. So you might actually get a data set, everybody's psyched, oh, new genotype data here, and you run, and you're like, nothing replicates, there's something wrong. You have to go to everybody and say, like, hey man, there's something really wrong with our data, which is like, I've been in that position a few times, and it's not fun, but everybody would rather know than not know, right? So anyways, that again, that's in our package. It's not in CRAN, and I wrote that code, so it works, but it's a little bit dicey. Oh, I guess, yeah, we're not going to do that. Like I said, I wanted to save time for questions, so we have 10 minutes if anybody wants to ask anything about Phecodes. And since I glossed over some of the details of some parts, I can always kind of roll back and explain anything else. But that's all I have to say.